Thank you for joining us today for our webinar. We're going to go ahead and get started. Evan, our BD Director, will kick us off.
Director here at Phosphorex. Welcome, and thank you for joining us today to our webinar titled, The Right Microfluidic Mixing Platform Plays a Crucial Role in Advancing LNP Clinical Development. The purpose of this webinar is to provide an overview of the LNP formulation process and review the empirical comparability of the three process mixing platforms utilized for formulation transfer and development here at Phosphorex. As a disclaimer, Phosphorex is an independent and technology-agnostic fee-for-service CDMO committed to offering clients the best suite of tools and operational options possible. As such, Phosphorex can assist clients to select and utilize the right platform for their specific project if an existing preference is not established. To introduce our speakers today, Dr. Nick Boylan and Sydney Marchando. Dr. Nick Boylan is Phosphorex's Director of Nucleic Acid Delivery, and here in Hopkinton, Massachusetts, Nick leads a team of scientists and engineers that specialize in the development and optimization of novel drug delivery systems, including lipid nanoparticles and polymer nanoparticles. Prior to joining Phosphorex, Nick directed the analytical and formulation development of a novel soft mist pulmonary inhaler while at Molecular Infusions, and contributed to the encapsulation of a wide variety of payload types in the Accurins proprietary nanomedicine platform at Bind Therapeutics. Nick holds a bachelor's degree in chemical and biomolecular engineering from Cornell University and a PhD in biomolecular engineering from Johns Hopkins University. Working with Dr. Nick Boylan is Sydney Marchando, one of our formulation scientists. Sydney joined the Phosphorex team in 2022 and beginning as a research and development intern before starting her current role as a formulation scientist. Sydney is a member of the nucleic acid delivery team, primarily working on the execution of formulation feasibility and optimization activities in the lab to support the development of lipid nanoparticles for the delivery of nucleic acid-based cargoes. Sydney holds an MS in biomedical engineering from Boston University, where she investigated targeted biomaterials and nanoparticles for women's health and hormonal applications. She also holds a BS in chemical engineering from Colorado School of Mines, where she conducted research on the use of biomaterials and hydrogels to address pediatric growth plate injuries. Before we start, I'll just provide a brief overview of our company here at Phosphorex. So Phosphorex is a fee-for-service CDMO specializing in particle-based drug delivery solutions. We're located here in Hopkinton, Massachusetts, and our state-of-the-art facility spans 30,000 square feet, with an additional 12,000 square feet of cGMP space coming online in late 2024. Our team of technical experts has approximately 2 decades of experience, offering comprehensive services from formulation feasibility and optimization to process development, scale-up, and tox batches. Phosphorex's core areas of expertise are lipid nanoparticles for nucleic acid delivery, polymer nanoparticles for targeted delivery, and polymeric microspheres for sustained release delivery. Our mission here at Phosphorex is to accelerate client programs by delivering a tailored service experience that's as precise as the science. I now introduce Dr. Nick Boylan to kick off today's presentation.
Thank you for the introduction, Evan. Today we'll be discussing the selection of the right mixing platform for lipid nanoparticle development towards the clinic. We'll start with some background on LNP formulations and the processes we utilize at Phosphorex. Then we will discuss in detail the 3 main mixing technologies we utilize in-house, which include the automated nanoparticle system from Particleworks, which is now part of Unchained Labs, the Ignite and Blaze systems from Precision Nanosystems, and our in-house T-Mixer. Lipid nanoparticles, or LNPs, are the leading non-viral delivery system. The formulations can be tailored to accommodate a wide variety of payload types, including siRNA, antisense oligonucleotides, messenger RNA, and plasmid DNA. Each unique cargo plays an important role in the consideration of the fabrication methods and overall formulation. Based on the success of the COVID-19 vaccines, LNPs are currently being tested clinically across a broad range of diseases, including infectious disease, cancer, and rare diseases. Lipid nanoparticles are formed through a self-assembly process. Starting on the left side of the diagram, we have our starting solutions, which consist of lipids dissolved in ethanol and the cargo prepared separately in an acidic aqueous buffer. These solutions are introduced into a microfluidic mixing device, and upon mixing, triggers the LNPs to self-assemble due to the decreased solubility of the lipids in the presence of water and the encapsulation of the nucleic acid cargo via ionic interactions with the positively charged ionizable lipids. Subsequently, the LNPs are purified by downstream processing, such as dialysis, to remove residual ethanol and exchange the buffer salts and adjust the pH. And this results in the final LNP structure depicted on the right side of the figure. So now I'll hand it over to my colleague Sydney, who will discuss our target product profile and walk us through the various mixing technologies we utilize here at Phosphorex.
Thanks, Nick. For this specific study, we targeted the following product profile. Utilizing poly A as our model cargo, we targeted a final particle size between 80 and 120 nanometers, polydispersity, or PDI, below 0.2, and an encapsulation efficiency of 85% or greater. We also evaluated the stability of the LNPs post-1 freeze-thaw cycle at negative 80 degrees Celsius. For these formulations, we utilized the ionizable lipid MC3 with its structure shown here on the right. This lipid carries a positive charge at pH 3, but is neutral at pH 7.4, so these factors played a role in the formulation design. We utilized a 4-component lipid system, including MC3 at 50 mol%, cholesterol at 38 mol%, the helper lipid DSPC at 10 mol%, and DMG PEG 2000 at 1.5 mol%. These formulations have a lipid to mRNA weight ratio of approximately 20, which equates to an N to P ratio of around 6. In terms of processing, we utilized a range of total flow rates between 40 and 90 milliliters per minute, and a constant flow rate ratio between the cargo and lipid solutions of 3. For mixing geometries, a variety were tested with this specific formulation, and we'll go into more detail on the results from each of these platforms. We worked with a droplet junction chip on Particleworks, now Unchained Labs automated nanoparticle system, the next-gen and next-gen 500 mixing technologies on Precision Nanosystems, Ignite Plus, and Blaze systems, and an internal system with a mixing T-junction. For downstream processing, small-scale batches underwent dialysis and centrifugal concentration, while larger batches underwent tangential flow filtration. All of the processes ended with 0.2 micron filtration and dilution with sucrose before being tested for freeze-thaw stability. This graphic provides an overview of the general process flow. After preparation of the cargo and lipid solutions, the particles are mixed via one of the 3 mixing methods before undergoing dialysis, centrifugal concentration, filtration, and dilution. For large-scale batches, TFF is utilized as a scalable unit operation in place of the dialysis and concentration steps. As I mentioned before, 3 different platforms were studied based on the 3 most common systems we utilize in-house for fabrication of lipid nanoparticles. On the left is the droplet junction chip, which is utilized with the automated nanoparticle system made by Particleworks, which is now part of Unchained Labs. The cargo solution enters the chip via 2 separate inlets, which intercept on either side of the central lipid line. The streams mix at the junction and then proceed downstream to be collected for further downstream processing. The second mixing technology is the next-gen technology for Precision Nanosystems. The Ignite Plus is the smaller scale device, which can utilize both the next-gen or next-gen 500 mixing geometries. The next-gen 500 geometry has slightly larger channel sizes than the next-gen chips. It can accommodate greater flow rates, and it's the mixing technology that's featured here in this image. The next-gen 500 mixing technology is also utilized on the Blaze system, which accommodates the larger scale batches. And then finally, the third mixing technology is our in-house T-mixer, which brings the cargo and lipid solutions together at a head-on T-junction. Now we'll discuss the first nanoparticle fabrication system, which is from Particleworks, now Unchained Labs. For this system, the automated nanoparticle system, or the LNP, we generated batches ranging in scale from 0.2 milligrams of cargo all the way up to 100 milligrams of cargo, which resulted in batches ranging from 2 milliliters to 1 liter of total volume. We also tested total flow rates ranging from 4 to 12 milliliters per minute through the microfluidic chips utilized on the system. For these batches, we used a 190 micron droplet junction chip. With this microfluidic chip, the cargo and lipid solutions are injected into the sample valves. The driver fluids, which are either aqueous buffer or ethanol, are utilized to then push the injected cargo and lipids into their respective slots on the microfluidic chip, where then they meet, and then the particles are then pushed out to the fraction collector, which enables a heads and tails cut to be taken at the start and finish of formulation, and then the particles are collected for downstream processing. So now looking at some of the results from the small scale studies on the ANP system, we looked at the 0.5 milligram scale, and particles were fabricated with varying total flow rates ranging from 4 milliliters per minute up to 12 milliliters per minute to test the impact of total flow rate on particle size, PDI, and encapsulation efficiency at this small scale. Overall, the particles fabricated at 4 milliliters per minute were larger in size and PDI than those fabricated at 8 milliliters per minute or 12 milliliters per minute at every downstream processing step, as well as for those final particles. But there wasn't a difference in encapsulation efficiency regardless of the total flow rate, and they were all able to achieve the desired target product profile value of greater than 85%. So based on these results, and after the investigation at the small scale, a total flow rate of 12 milliliters per minute was selected for the scale-up efforts in order to achieve those desired particle parameters, but also to minimize fabrication time based on the larger flow rate. So looking at the scale-up, we were able to use 12 milliliters per minute at the 0.2 and 0.5 milligram scales, and then scale all the way up to 10 milligrams and 100 milligrams of cargo. For the small scales, we used dialysis and centrifugal concentration for downstream processing, and for the larger scales, we utilized TFF for the downstream processing. Overall, with the scale-up efforts, the lipid nanoparticles made across the range of scales were similar in size and PDI, and all achieved the desired target product profile. And also the encapsulation efficiency was similar across the batch scale, regardless of the size of the batch. Additionally, looking at a freeze-thaw cycle, we were able to maintain the stability of these particles after a freeze-thaw at negative 80 degrees Celsius, with similar particle size, PDI, and encapsulation efficiency at all of the scales tested before and after the freeze-thaw. So overall, we were able to successfully scale this formulation on the ANP system using a total flow rate of 12 milliliters per minute. The second system is the Precision Nanosystem instruments, which utilize single-use chips with the next-gen mixing technology. For the Ignite Plus system for small scales, we tested scales ranging from 0.1 milligrams up to 4 milligrams of cargo. The Ignite Plus system can use both next-gen and next-gen 500 chips. So we tested flow rates from 4 to 16 milliliters per minute on the next-gen chips and 90 milliliters per minute on the next-gen 500 chips. The next-gen 500 chips then scale to the Blaze system. So we tested 90 milliliters per minute again on the Blaze system, looking at the 10 milligram and the 100 milligram batch scales. The Ignite Plus system has entirely single-use flow paths, while the Blaze system uses a single-use mixing cartridge with upstream reusable fluid paths for the larger input volumes. So looking at the small scale, we again looked at the impact of the total flow rate on the size and PDI at the 0.1 milligram scale. Overall, we tested flow rates ranging from 4 to 16 milliliters per minute using a next-gen chip. Similar to the ANP system, trends with the highest particle size and PDI at the lowest flow rate of 4 milliliters per minute, and then similar size with increasing total flow rate for the 8, 12, and 16 milliliter per minute conditions. Overall, the encapsulation efficiency was consistent across the total flow rates and was well above the goal of 85% or higher, regardless of the total flow rate. With scale up to 100 milligrams, the next-gen 500 chips were utilized on the Ignite Plus system for the 1 milligram and 4 milligram scales, and on the Blaze system for the 10 milligram and 100 milligram scales. A flow rate of 90 milliliters per minute was used on both of these devices for fabrication. Overall, the particles generated at each batch scale, regardless of the system used or the downstream processing method, were similar in final size and PDI. And additionally, with looking at the freeze-thaw, the size, PDI, and encapsulation efficiency was maintained before and after the freeze-thaw stability was conducted. So overall, we were able to scale using the NextGen 500 chips on the Ignite Plus system to the Blaze system for a final batch size of 100 milligrams. The third system is our internal mixing platform, which utilizes a mixing T-junction to combine the cargo and lipid solutions head-on. This system's operated at the small scale, so the 1 milligram scale, using syringe pumps. And then at the larger scales, it's using the L-flow system, which enables the pressure-driven flow of our cargo and lipid solutions. So we used the 1 milligram scale for small scale studies here, and then we scaled up to 10 milligrams and 100 milligrams on the T-mixer. At the small scale of 1 milligram, we looked again at the total flow rates impact, using flow rates from 16 milliliters per minute up to 32 milliliters per minute. At this scale, there was no impact of the increasing flow rate on the particle size or PDI, with consistency throughout the downstream processing regardless of total flow rate, and similar final particle size and PDI. Additionally, the encapsulation efficiency was consistent regardless of the total flow rate at this small scale using the T-mixer. And so again, based on the parameters that we desired for the particles and also taking into consideration the total time of fabrication, we selected 32 milliliters per minute for the large scale to scale up from 1 milligram to 100 milligrams. At this scale up step, there was good comparability of size and PDI across the scales. And overall, it was similar for the encapsulation efficiency as well. Additionally, looking at the freeze-thaw stability, it was again maintained before and after the freeze-thaw at all three scales tested with the T-mixer. And so we were able to successfully scale this formulation as well on the T-mixer from the 1 milligram scale on the syringe pumps up to the 100 milligram scales using the L-flow system. For these studies, we largely looked at the size and polydispersity using dynamic light scattering, and the final size ranged from 80 to 120 nanometers with a PDI less than 0.2 for the three different mixing platforms. Additionally, encapsulation efficiency and concentration were tested using ribogreen, and for all of these formulations, the encapsulation efficiency was greater than the target of 85%. We additionally tested for the freeze-thaw stability at minus 80 degrees Celsius. In-house, we also have additional analytical capabilities, including zeta potential by laser Doppler electrophoresis, lipid quantitation, and the capacity for automated ribogreen assays, as well as other internal capabilities or other additional analytical capabilities available through a third party. And so in summary, Phosphorex offers a flexible range of microfluidic mixing technologies to best accommodate our partners' LNP development path towards the clinic. In this study specifically, we demonstrated how the microfluidic mixing step was optimized at the small scale to achieve the target product profile of size, PDI, and encapsulation efficiency across three different mixing platforms that we utilize. These platforms include the Particleworks Unchained Labs automated nanoparticle system, the Precision Nanosystems Ignite Plus and Blaze platforms, and our internal T-mixer system. Overall, the optimized conditions were then successfully scaled to 100 milligrams, and acceptable freeze-thaw stability was achieved with regards to the size, PDI, and encapsulation efficiency for all three mixing platforms. In addition, Phosphorex has additional formulation optimization expertise across a wide range of nanoparticle-based therapeutics.
Thank you, Cindy and Nick, for the presentation. See that you're already on screen. Make sure you're on mute. We're ready for the Q&A session. We've had a couple of questions that has come in, which I believe Nick has been tracking. But Nick, did you want to kick us off with the first question?
Yeah, thank you, Kim. And also like to give a special thanks to our participants today for joining us and for submitting a number of great questions here. So Cindy and I can take a few minutes to answer some of these, but if in the future you have any questions or want to further inquire about Phosphorex capabilities, feel free to email at us at the contacts listed here. So the first question we have here is regarding the target product profile. And in particular, why did we target a particle size of 80 to 120 nanometers? And so in this case, this happens to be the most common particle size range for these LNP products. One other consideration as well is that these products are commonly terminal sterile filtered, and therefore having a size below 200 nanometers is important to ensure that you have good yield or recovery of the batch after passing through a 0.2 micron filter. And then there's also a popular dogma that suggests that maybe particles, the smaller the better. So therefore, potentially, an LNP that's formulated at 80 to 120 nanometers might be superior in terms of potency in vivo as compared to a larger LNP. But again, I believe that's sort of a dogma in this space. So therefore, I wouldn't rule out the possibility of exploring particles that are larger. But one challenge might be ensuring that final product is sterile. But yeah, certainly a great question there. So the second question we have is, did you test different channel structures from Particleworks? And did you basically have a preference, right? So did the droplet junction chip work better than other chips, for example? Yeah, so Sydney, would you like to cover that one?
Yeah, I can discuss this a little bit. So we've primarily used the droplet junction chip with the 190 micron channel size. And it's worked well for us in most applications that we've used it for. With the AMP system, there is a micro mixer option, but we've had some challenges keeping this chip clean and free of foreign debris, just compared to the droplet junction chip with the more complicated flow path, making that cleaning protocol just a little bit more challenging. So kind of today, we primarily do use the 190 micron. In the future, we are looking to explore some of the larger ones, such as the 275 micron droplet junction chip, for formulations or challenging formulations that are more prone to problems with chip clogging. There's also a 5-input chip that we're hoping to do some similar investigations on as well.
Yep. Yeah, thanks, Sydney. And so the next question here is, does the Particleworks instrument support high throughput formulation capabilities? And coupled with that, or is it one of the formulation at a time, as is the case with Ignite? So I can comment on this really quick. So with the AMP system, it does have some built-in automation through the use of what they call the protocol mode in the software. So with this feature, you can effectively inject one solution of cargo and one solution of lipids into sample injection loops. And then the system can automate the injection of those solutions through the microfluidic chip. So within a single run, you can vary the total flow rate and flow rate ratio and effectively produce multiple batches. So again, that is the automation. That is a nice feature there. And kind of combined with that, the system will perform automated cleaning runs between batches, right, to minimize kind of cross-contamination. And then likewise, at the end of that protocol mode run, it will do a more intensive cleaning procedure. However, yeah, if you're looking to perform higher throughput studies, their newer Alice system would really be the preferred format, in which case you could effectively load 96 well plates of your lipids and cargo solutions. And then that system is designed to be more kind of a higher throughput, right? So you could, formulate formulations up to 96 different formulations at once with that system. So, and that's certainly, a system that has been getting a lot of attention in this space due to those added capabilities there. Yeah, so excellent question there. So the next question is, can you comment on the post-processing stability of the nanoparticles, and how about the operating and storage temperature? So after doing the mixing step, the formulations are effectively suspended in 25% ethanol, likewise in a slightly acidic aqueous buffer. And therefore, it's generally desired to exchange that buffer for one that is more neutral, right? And also in the absence of that ethanol. And then this will effectively kind of stabilize the LNP particle. And furthermore, depending maybe on the ionizable lipid, maybe there could be some degradation, right, at those lower pHs. So again, one other reason why you want to kind of minimize that hold time if possible. However, you would really need to look at each specific formulation, what that stability is post-mixing. And so one routine study that we'll do is, after maybe performing microfluidic mixing on a particular formulation, we can take that suspension, and in these cases, it's often just at room temperature, but we might hold that suspension, right, for a period of time, maybe up to an hour. And then over that course of that hour, we might sample and measure the particle size, for example. And if we're observing very consistent particle size, then at least that would give us, some indication that the formulation is handling those conditions fairly well and is being fairly stable. Or alternatively, if the particle size was rapidly increasing, right, over the course of 15, 30 minutes, that would certainly highlight the need to one, either minimize the hold time after the microfluidic mixing, and or maybe to add in a dilution step. So like inline dilution could be performed, right, to more quickly transition from, again, that initial acidic environment to a more neutral environment. So yeah, definitely a great question there. So for the next question, maybe I'll hand it over to Sydney, but it's what is the internal diameter of the T mixer?
Yeah, I can take this one. The internal diameter of the T mixer is 200 microns, so slightly larger than the 190 micron diameter chip. Oh, and I guess, yeah, we also have a 500 micron option as well, but the 200 is what we utilized for these studies.
Yeah, thanks, Sydney. And let's see. So the next question here is, does the size of the cargo affect the encapsulation efficiency? Yeah, I think this is a really good question. I think kind of to date, with the size ranges of the cargoes we've experienced, we haven't necessarily seen a direct impact. Like, so for example, maybe larger cargoes having a lower encapsulation efficiency. I think the one parameter that kind of comes into mind is the ratio of the lipid to cargo, and this is frequently reported either on a mass basis, or it might be reported on a molar basis in the form of the nitrogen to phosphate ratio. But for the formulation presented here, we had the lipid to cargo mass ratio of 20, right? So we have basically 20 times excess mass of lipid. And so my expectation, right, if we looked at a range of cargoes, maybe going from 1,000 nucleotides up to 10,000, as long as we're maintaining that mass ratio, I would expect very similar encapsulation efficiencies. However, even with a single cargo, if we start playing with that mass ratio and maybe using less lipid, I do anticipate at some point we might see a lower encapsulation efficiency. So that's maybe one comment there. And then I think secondly as well is related just to the ionizable lipid itself. So certainly depending on the structure and also maybe the pKa of the ionizable group, that can directly impact its ability to efficiently encapsulate those cargoes. Yes, certainly a good question there. Let's see. So the next question, in addition to LNP quality attributes, did you look at delivery efficiency and cargo expression rates in cells? And so, yeah, I can comment on that one quickly. So kind of to date, we haven't performed like a systematic side-by-side comparison. This is something that, yeah, we are very interested in doing and hope to do in the near future. This would require us to transition from the poly A cargo that we utilized here today to a messenger RNA that has a convenient reporter protein. So maybe like a human EPO, or even better would be like a luciferase, for example. However, what we can say is that kind of to date, we have successfully, transferred formulations. Sorry, maybe I'll end that one there. So for the next question, to date, have we directly compared the potency of LNPs prepared on the different mixing platforms? My bad. Sorry, I'm getting a little confused as we have kind of multiple questions in front of us here. So I'm gonna jump to the next question. Maybe when my brain catches up, I might be able to circle back on that one. So for the next question, can you comment on the size increase observed post dialysis? So here, what you may have noticed during the slide presentation is that we'll often observe a size increase post manufacturing and then after going into dialysis. And so oftentimes this represents sort of a ripening process that the LNPs exhibit. And so again, after performing the microfluidic mixing, the LNPs are suspended in that acidic buffer with 25% ethanol. But after doing that downstream processing by dialysis, right, we've effectively removed that ethanol and positioned the particles into that neutral buffer. So it's during this process that the LNPs may kind of structurally have some changes, right? That is likely what is causing the exhibited size increase. This isn't something that we've necessarily looked into in great detail with regards to cryo-EM, but I think that could be a very interesting study to conduct in the future. And then kind of just related to this observation as well is basically when we subject particles to the tangential flow filtration process, we oftentimes also observe a slight increase in particle size relative to those that were only subjected to dialysis. And just to kind of dig into that a little bit, essentially when you're performing tangential flow filtration or TFF, you're utilizing a peristaltic pump to effectively flow your LNP solution through a semi-permeable membrane, being the TFF column. And so this subjects the particles to additional stresses in the form of shear stress, for example, in both the pump head and likewise in the TFF membranes. So again, this additional stress can cause the particle size to increase, and that is something that we commonly observe. However, by having the appropriate kind of controls in place and the appropriate optimization of the TFF kind of process, you can try to minimize those size increases. Okay, and so for the next question, maybe I'll hand it over to Sydney for this one. But what is the stability of LNPs post microfluidic mixing?
Yeah, I'm happy to take this one. And I think we discussed it a tiny bit earlier, but it really is formulation dependent, and a lot of factors can play a role in that. Some of that being kind of the scale of the batch and how long the holdup time after is, and how long those intermediate suspensions are held at various conditions. And so, yeah, testing the kind of the time dependence of size and seeing if there's changes is a great way to monitor the stability of those kind of throughout downstream processing and see changes that occur to size, PDI, et cetera with that. And then I think, as we mentioned, kind of incorporating maybe inline dilution can help with that process. And one downside to that is you do end up with larger process volumes. So it does have an increase of kind of the formulation time after that of reconcentrating either via TFF or a different concentration method once you've run the inline dilution, but it has, yeah, that is one way to address those stability concerns if you do see any kind of inconsistency or changing in size with time throughout the downstream processing.
Sure. Yeah, thanks, Cindy. Maybe one more for you would be, what will Phosphorex evaluate in future experiments on the 3 mixing platforms?
Yeah, absolutely. Also happy to take this one. So kind of beyond what we looked at here, which was really the size, PDI, encapsulation efficiency between the 3 processes, we definitely are really interested in looking at how these 3 different mixing options might play a role in the actual LNP particle morphology. So being able to look at the particles and see what the actual morphology of those looks like compared to just the size that we get via DLS. And so that's definitely something that we're interested in doing in the near future, hopefully, and looking at the morphology with a method such as cryo-TEM.
Okay, perfect. Yeah, thanks, Sydney. So another question I see here is, are there any differences in encapsulation efficiency between DNA and RNA cargos? So that's the kind of the first part of the question. And so for that, I would say we frequently see very similar encapsulation efficiencies. However, we will frequently see a difference in particle size. So I think encapsulation of, say, a plasmid DNA, especially if it's larger than the messenger RNA, we'll tend to see a larger particle size associated with that DNA cargo. And then one other consideration there as well is, for plasmid DNA, it tends to be in a supercoiled state. And that's the preferred kind of format for the plasmid DNA, right? Once it's encapsulated, and you want to maintain that the supercoiling. So really the stability of that plasmid DNA is also important, that you want to verify, right? Starting with the supercoiled DNA, that upon encapsulating and subsequent release, that you're maintaining those properties, and inherently you're not accidentally maybe nicking that DNA and allowing it to basically uncoil into a more linear form, for example. And then kind of the second question from there is, are there human tissue specific LNP formulations? And I certainly see that as kind of like the next frontier, right, in the LNP space. So certainly we have a lot of lipid systems that are very efficient at, going to the liver, right, and kind of expressing cargoes and hepatocytes. And certainly a lot of work in the literature kind of exploring formulations, right, that can target other tissues and organs. So I believe, right, we're essentially on that path. And I know there are definitely a few kind of general approaches that can be utilized. So one is kind of playing with and in this case, I'll just call it the overall charge associated with the lipid nanoparticle. So with your most common ionizable lipids, they tend to have, maybe for hepatic delivery, right, they have a slightly, pK maybe around, let's call it 7. So therefore at neutral pH, these tend to have a zeta potential or surface charge that's just slightly negative. However, if you were to, in this case, maybe you formulate with a cationic lipid that's positively charged, right? And now that particle is going to have a much more positive surface charge. That in itself can definitely impact where in the body it distributes. In a lot of cases, it might actually distribute more towards the lung versus the liver in that particular case. So again, that's one method or approach that can be utilized, is playing with, let's call it the pKa of the ionizable lipid, or maybe other lipid components you incorporate. So whether it's a neutral lipid, negative charge, positive charge will impact how they distribute in vivo. And then really maybe a second approach as well, right, would be if you conjugate to the particle surface, maybe a ligand, right, that is specific for a certain cell type. That might be one other approach. So using active targeting to facilitate, maybe you're targeting immune cells in this case, for example. So I think, there's certainly a lot of kind of interest, right, in work that's being performed in that space. So, and personally, it's something I'm looking forward to just seeing as the field kind of advances. Okay, so one of the next questions we have is, what type of TFF pump system is ideal for LNP TFF processes? And do you see issues using the peristaltic pumps? And so, what I can comment there really quickly is we do have just your common peristaltic pumps. We have Repligen, both our KR2I system for small scale batches, and that's the system we use, excuse me, primarily with our LNP batches. And we also have a larger scale system, KMPI system. And we do successfully utilize those pump systems. However, there are these additional pumps that do subject the suspension to less shear. And so I believe these are the Lebtronic pump systems. So in this case, so whereas with a peristaltic pump, the pump head actually makes a physical contact with the tubing, which contains the LNPs. With these Lebtronic pumps, the impeller is basically suspended using magnets, and therefore creates less shear stress on the LNP suspension. So I know that is one alternative pump system that we've looked at internally, and does appear to be promising in terms of kind of controlling how much shear those particles are subjected to. So definitely great question. So bear with me as a second as I kind of look through some of these additional questions in the chat. Yeah, and I think kind of coupled with that question was, the material type with TFF. Yeah, and then, yeah, maybe this is something we could go into maybe in a future webinar. Honestly, kind of to date, we have primarily used the modified PES membranes in the hollow fiber format. However, we are starting to look into flat sheet membranes, right? And there's also regenerated cellulose. So I do think this is an area worth kind of investigating, right? Is kind of, what is the preferred TFF membrane format for basically doing that downstream processing, being the buffer exchange and concentration of these LNPs. And again, we have done some work in that. But most frequently, again, we're using the modified PES in the hollow fiber format, and those do tend to work well for us. But I do suspect, right, there can be some further optimization there. And yeah, off the top of my head, yeah, I won't go into further details on the TFF, but yeah, certainly, for those that are interested in those additional details, yeah, if you kind of reach out to us by email, we'd be happy to set up a conversation to discuss those in more detail. And so one other question we have here is that I can address is, in addition to the total flow rate, the flow rate ratio, and chip geometries, are there other critical process parameters to be mindful of? And really kind of the one example I'll give here is, if your system enables this, you're trying to monitor the operating pressure of the system. So this is a good way to ensure that you have the same operating conditions, right, from batch to batch, and good batch reproducibility. So when this microfluidic process is running at steady state, you should have very consistent pressure in both your lipid and cargo feed solutions. And similarly, as you kind of scale up, those pressures should be maintained. Really, some red flags that could be observed in any variation in the pressure would be if the pressure is increasing over time. This might indicate that maybe the microfluidic chip is being fouled over time, right? So maybe some lipids are depositing. And at some point, that might actually completely obstruct the flow path and result in batch failure, right? So this is certainly something you don't want to occur when you're manufacturing an important batch at large scale. But again, actively monitoring these, at small scale, you'll have a good kind of foundation of, how the process operates. And this is very easy to monitor on the A&P system because it does display the pressure of the pump systems. And also the blaze system as well. And then likewise, yeah, if you were observing a lot of feed pressure fluctuation, this could also be indicative of other problems in the system. So for example, maybe there's a leak somewhere, or likewise, if you're using syringe pumps, if one of the seals on those plungers is failing, that also can result in some inconsistent pressures. Okay, so the next question we have here is, what is the size of the poly A cargo? And this one, I'll hand it over to you, Sydney.
Yeah. So the poly A that we use is commercially available and by gel electrophoresis has a size of around 6,000 nucleotides. And this is generally larger than the typical mRNA cargos that we've utilized. So luciferase has a size of around 2,000, EGFP around 1,000, same with human EPO, also around 1,000. And so, yeah, the poly that we use is a little bit larger than those standard ones. And then for comparison, the Cas9 mRNA has a size around 4,500 nucleotides. So we've noticed, just kind of based on our historical data, there might be a little bit of a size impact on the final LNPs based on that, the size of the cargo that we're using, but kind of to date, we haven't seen too large of a difference between, just for example, a particle made with the EPO mRNA versus a particle made with the poly A as the cargo, very similar in size.
Okay. Yeah, thanks, Sydney. And maybe for this next one, if you could address as well. So does Phosphorex have experience with other lipid systems?
Yeah, I can address this. Yeah, so in addition to working with MC3, we do have experience with a couple different lipid types, including the ALK0315 from the Pfizer-BioNTech COVID vaccine, and then also Moderna's SM102 lipids. We've also worked with other ionizable lipid systems that have been reported in the literature and are available for R&D purposes, and so we do have experience with those. We also have experience with screening lipid libraries that are designed by our partners and looking at formulations with those, and also with beyond the ionizable lipid, looking at helper lipids, selection of cholesterol derivatives, different PEG alternatives.
Okay. And
Just
for the,
one last question, Nick.
yeah,
If
thank
that's
you,
okay.
Kim.
Yeah, we need to wrap up. Thank you.
Yep. Yeah, and for the final question here is, can you compare and contrast the various mixing systems? So Sydney, if you want to kick us off on that one.
Yeah, I can start us off. I think kind of overall, there's definitely attributes of each of the three systems that we talked about that are unique to the instrument. A big one being that mixing geometry. So each system kind of does have a unique mixing junction, whether that's the ANP system or the PNI systems or our T-mixer, unique in how it brings those cargo and lipid lines together. Also just kind of with the interaction with the system and the operation with how the systems drive the fluid. So whether it's pressure driven or syringe pump driven, just, yeah, slight differences in kind of that interaction, but overall they, yeah, kind of have a similar approach, I'd say, to the being able to fabricate similar scales of fabrication feasibility in terms of small scale to large scale. So big difference being kind of your interaction with the system, the software of the system, and then also that mixing microfluidic junction that brings those lines together.
Okay. Yeah, thanks, Cindy. Maybe I'll just kind of wrap up this one in that I think we're kind of privileged to have all these kind of internally right here at Phosphorex. So we get familiar with all the platforms, and yeah, we've had good success, right, transitioning formulations between them, and yeah, they each have kind of the unique benefits, right? So we can kind of, for a particular program, we can kind of help select, which platform to utilize. Excuse me. And in general, when we're working on a partner program, oftentimes it's maybe preferred to use the same platform, right? So if we have a partner that's using, let's say, the PNI Ignite, right, we can use that same mixing technology in-house, or alternatively, right, if somebody's using Unchained ANP system, right, we can maintain that same operating system internally. So thanks again, everyone. Maybe I'll hand it back to you, Kim.
Thank you, Nick and Sydney, for really great presentation and a long Q&A session. I apologize about the screening glitching back and forth. The mouse is a bit more sensitive than we had thought. But thank you, everyone, the audience, for joining us today. But a recording of this webinar will be sent to you via email within 24 hours or so. Have a great rest of the day. And here's our contact info. If you would like to reach out directly to Sydney, Nicholas, or even Evan, please feel free to do so. And we are going to wrap up. Thanks again for joining us today. Have a good day.
Thank you. Thank you, everyone.
Thanks, everyone.